Hello everyone and welcome to CK Med. My name is Clark and I'll be taking you through the development of the reproductive tract. Now keep in mind that this is not that entertaining, but I feel that this is a great way of remembering some of the structures, some of the way that things develop, and some of the disorders that come from this. That is actually super high yield for reproductive questions on your step 1 and can come up for your anatomy course. Because in embryology, they give you slides and slides of pictures and pictures and pictures with 500 things labeled on every single one of those pictures. It's very hard to see what's the big picture that's actually happening, what's going on, what things are very important to know that develop into certain other things. So I'm here to kind of discuss that, walk you through what's important, what's not important, and things to kind of focus on. All right, so to start things off, before we actually get to anatomical structures or embryological structures, you need to understand the physiology and genetics that actually kind of come into play in developing a male versus female. And so it really starts down at the basics of cells. And so I know it says medulla and cortex, don't worry about that, but the first thing you need to start off with are XY versus XX chromosomes. And so the big thing is that on Y chromosomes, you have a gene called SRY. And so if you're sorry, then you make testes. If you're not sorry, then you make ovaries. That's the easiest way to kind of remember this. So the starting point is if there's a present SRY gene, then you make testicles, and that means you make Leydig cells, you make Sertoli cells, and from that point on, then you can go ahead and develop all the internal other structures within testicles and start making all the other hormones and whatnot. So if you're sorry, you make testicles. If you're not sorry, you make ovaries. We will come back to the female side in just a minute, and we'll spend more time on that a little bit later. But as far as the male side, if we are sorry and we make testicles, that means we make Leydig cells. And Leydig cells are important for making testosterone. We'll come back to that. And we also make Sertoli cells, which are important during development for the production of Mullerian inhibiting factor. That's the only thing we need to worry about at this point in time. And what Mullerian inhibiting factor, it means in a basic embryo, before X and Y and XX get kind of expressed to develop male versus female, we have both male and female parts. Now, if I turn on and promote one part, then I need to turn off the other, and that is when we have our Mullerian inhibiting factor. So this turns off the female part of its internal genital tract. Now, this is where number 1 on this picture that first aid has emphasized is pointing out is that if we're lacking Sertoli cells or they are non-functional during the development of this, yes, we have already made testicles. Testicles are there, they're in the abdomen, ta-da, right? We have testicles now, and they might be missing Sertoli cells within those testicles, but we still have testicles. So as long as we reach this level, we have testes. Now, if we want to get to this level and have testosterone and Mullerian inhibiting factor allow for normal production, then we need both of those cells, the Leydig cells and Sertoli cells, to be working. If they're not, and we don't produce Mullerian inhibiting factor, then we are not going to inhibit the internal female genitalia production. If we do, then we do inhibit the internal female genitalia production. So say everything's normal. SRY, we're sorry, so we make testicles. In the testicles, they make Leydig cells and Sertoli cells. We make Mullerian inhibiting factor. It turns off the female pieces and makes testosterone. Now, testosterone, what it does is it goes and produces and promotes, it upregulates the production and growth of the male parts, the internal male genitalia. So that's very important. In addition, if I am producing testosterone, that testosterone will be converted from an enzyme called 5-alpha reductase. If you go to your steroid hormone and biochemistry, you'll see this enzyme changing testosterone into DHT, or dihydrotestosterone, or DHT. Now, DHT is special. It does everything that lets you look at a man and go, you look like a man. That means it gives you lots of hair, it causes male pattern baldness, it gives you a penis, a scrotum, and if you were to stick their finger up their butt, you can feel for prostate. And so DHT is very important for producing those structures on kind of physical exam, inspection, as well as palpation of the prostate. Okay, all those things are male external kind of features. Where internal stuff, more like seminal vesicles, epididymis, ejaculatory duct, and ductus deferens, all those tubular structures, are things that are promoted from testosterone alone, not converted to DHT. So testosterone converts these guys here. So what happened? We had our XXY, the Y chromosome created us to be sorry. We made testicles, Sertoli cells, and Leydig cells. We inhibited the female stuff. We promoted the production of internal stuff with testosterone, external stuff with DHT, and if we were to cut off this enzyme deficiency of 5-alpha reductase, you can see that we can still make male inside stuff. We just can't do outside. So we'd have sort of like an ambiguous genitalia for that, or we might even have female-like genitalia because we weren't able to make DHT if 5-alpha reductase is the deficiency. Now, in female, on the other hand, if we're not sorry in the XX chromosome, no SRY gene, then we make ovaries. And we don't make Leydig cells, and we don't make Sertoli cells, we just make the follicles, and we make all our other cells found within our ovary, and that will eventually lead to the production of estradiol, which is an estrogen. Now, estradiol is the thing that does all the external genitalia. Just like DHT was the one that made external stuff over here, estrogen is the one that does the external stuff over here, and the lack of Müllerian inhibitory factor is all you need in order to produce internal genitalia. So let's talk about some of these different disorders. There's one other one that you should be able to recognize just from this physiology, is what happens if I actually have, this is a receptor found right here, that DHT kind of comes in and binds a receptor and makes external stuff. But what if this receptor is faulty, so faulty receptor? Then this leads to androgen insensitivity disorder, and this is something very interesting. Okay, so androgen insensitivity disorder is a male that doesn't look like a male. It actually looks like a female. And so how you actually see this is, remember, if we start up here, XY, it's an XY human. XY human makes SRY, makes testes, Sertoli cells, Leydig cells. Sertoli cells will inhibit this. We have no problem with that pathway, so no female stuff is produced. And Leydig cells make testosterone. Now, this is the pathway that's inhibited through DHT, so we are not going to make external male-like stuff, but there's no problem with testosterone binding and doing its internal stuff. And so this is actually an external-looking female with internal male genitalia, such as seminal vesicles, epididymis, ejaculatory duct, and ductus deferens, and they also have testicles. So this is the female with testicles, internal genitalia, but external genitalia of female. And so this is something like you can get those disorders that will show up on STEP and also in your physiology for repro section, so definitely be able to recognize that. All right, next is what is happening on the inside here. We talked about those in a few words, so what is actually happening, or what are we looking at? And so this is kind of, you have to identify kind of the basic structures here. So we have this gubernaculum, which is this pink thing. Don't worry about it, whatever. We talked about it earlier in our other videos, but as far as right now, don't worry about it. So we have this undifferentiated gonad, and we also have this mesonephric mesonephros and mesonephric duct, and we have this paramesonephric duct, which is the yellow here. Okay, so this yellow is female stuff, and this green is male stuff. And so just like we were talking about on the physiology before, if we kind of get rid of one and keep the other, or get rid of the other one and keep the other one, then we will get the associated internal organs of the female and male reproductive tracts. And this kind of shows what happens when we have no androgens, such no testosterone and DHT, versus DHT and testosterone together, so male versus female. So when we have testosterone and we have DHT and the receptors are totally working fine, we're making male stuff on the internal. So we're making male on the internal stuff, then we also are making from our Sertoli cells the Mullerian inhibitory factor, MIT, and that is destroying this yellow thing, which is our paramesonephric duct. And this allows for our testicles to be attached to its tubules, such as our vas deferens, and our epididymis is this part right here that's pointing to. And this allows for our testicles to be connected to this portion, which is our urethra. And your urethra then allows us to be ejaculating the sperm that comes from these organs. And then we also have this mesonephric duct, and this is part of the ureter, and that goes up to the bladder here, which is fine. Don't worry about that too much right now. But on the female side, when we don't have androgens, we don't have testosterone, we don't have DHT, and we do not have Sertoli cells producing Mullerian inhibitory factor, then all the female stuff is going to remain, and we're not going to promote the male stuff. So the male stuff will disappear, such as this one right here. The mesonephric duct will disappear, which is this right here. The paramesonephric duct is totally fine. Remember, all we had to do is not have Mullerian inhibitory factor, and this yellow will still live. Mullerian inhibitory factor is sort of like a poison for this yellow structure. If it's not there, it's still alive. And now this paramesonephric duct on the left side and on the right side are going to fuse together at the bottom, and they're going to make a uterus. So now you can see the ovary, we can see the fallopian tubes, and we can see this fused bottom. So imagine I have two tubes like this next to each other. La, 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 la. Okay, two tubes, two paramesonephric ducts. What happens if I stick these two together? Well, we get this largy, pouchy like thing. Similar if you took two tubes of Play-Doh and you stuck the ends together on one end. Well, I'd have this one thing. And so this is actually my uterus here, is what this is. So we're just kind of hooking these two things together at the end. And now at the end I have, my ovary, and the ovary, fallopian tubes, and my uterus. That's what my paramesonephric duct is doing. So if I were to describe what does the mesonephric duct do in men, the M of mesonephric ducts is for males structures. That makes seed, which is seminal vesicles, epididymis, ejaculatory duct, and ductus deferens. Everything except for the prostate. Remember we were talking about that develops from something else. That's from DHT promotion. Okay. Now our paramesonephric duct is for our females, and this creates fallopian tubes, uterus, and upper portions of the vagina, like we mentioned right here, upper vagina. Okay, and this is a nicer picture of this. So if I had two blue tubes hooked together, and then I scrunch them together, I'd see that I have one paramesonephric duct filling up this side, and then I have the other paramesonephric duct filling up this side, and we kind of fuse them together. Now I have my uterus, that is the actual shape of the uterus, and then the upper vagina, which is right here. Okay, and so that's kind of the female reproductive tract. All right, so now that was the internal. What about the external? What's happening here? So before our sorry or not sorry or go in play, the external genitalia look like this, undifferentiated. They're open, it's wide, there's a tubercle here that's going to either turn into the glans penis or the clitoris in a woman. And then either this is going to close up and seal up, and that's the ventral surface of the penis, or it's going to remain open, and that's kind of all your labia majora and minora found right here. And then there's a connection to the anus, which is over here. Don't worry about that, that's fine. Okay, so under the promotion of DHT, meaning that I have 5-alpha reductase working, I have my Sertoli cells that were inhibiting internal stuff, and I also have my Leydig cells producing that testosterone, which converted into my DHT. Now I can promote external structures for forming, and that will close off this structure found right here, my labial scrotal swelling and my urogenital fold. Those will close up and seal off this tube, and they will extend further and create a tube within here, and that's my urethra found within the genital tubercle, which is now called my glans penis. And this kind of seals like a zipper, zzz, all the way up between here, all the way down. And if this doesn't occur, if something goes wrong in this zipper, and so we have an opening either here on the scrotum or here on here, this is what we would call hypospadias, which we'll be describing in just a little bit. That's something that can occur. But if for some reason this urethra, the upper portion above the genital tubercle, if this gets misaligned or goes all haywire, then the upper portion or the dorsal surface, the top of the penis, actually will have an opening to the urethra, and that's called epispadias. So we'll be talking about that later as well. And then on the other side, if we have female and we don't actually zipper this up, we just keep it open and we actually open it up even more, then we have the vagina, which hooked up to your paramesonephric duct, which we talked about, and our opening of your urethra. This is where urethra, the bladder, can connect to and urinate out, and then the genital tubercle has resided and remained as a clitoris or a sensory point. Okay, so all these different structures down here are named, and they go to what on the male or female side. Okay, all right, so let's talk about some of the congenital abnormalities in both male and female on the next slide. So hypospadias, as I was talking about, in the scrotum or along the urethra, means urine comes out of the bottom, or the ventral, meaning belly of the penis. Remember, the anatomical position is an erect penis. So these are ventral areas. So this is hypospadias on ventral surface. Now what is happening here is a failure of the labioscrotal folds, or labioscrotal folds. And that is what we were pointing out last time, or on the last slide. It was going to be your labia majora and minora, but when they seal up and zipper up, it should be making a nice tube. If they don't and leave a little bit opening, that's hypospadias on the ventral surface. Okay, and that's as far as this. This is where your labia majora is going to be aligned here in the scrotum. But if it's actually on the urethra up here, then it's just fusion of the urethral fold actually failed. So that's important to know. And then epispadias on the other side is this one down here. Epispadias is where it opens up on the dorsal surface, on the back of the penis. And so this is due to a failure of the spongy urethra to form. Another way they could say that is, remember this tubercle, genital tubercle, is the thing that's kind of the column, and if it moves up or down, moves around, failure formation of that genital tubercle can actually cause epispadias as well. So either look for this definition or look for this definition when someone is described with epispadias. Okay, and on to one of our last things we're going to be talking about is our uterine congenital abnormalities. There's a whole bunch of them, but simply all you need to know is that when I mess up the uterus and I have two cavities, or they're not fused all together, or if I have like a little septum that comes down like this, it's due to the paramesonephric duct, like we talked about, a little Play-Doh columns not fusing again properly. And so if you have a uterus, a didelphys, or a double uterus, that's failure of complete fusion of the lower part of the paramesonephric duct. And so we left some of this tube separated from them, and now we have this kind of septum in between them, such as in this case as well. Like here you can see these little septums dividing. And so that is someone that has uterus didelphys, okay? And so, or like even two of them, right? So this is not fusion at all. You can see this right here. This is two uterus, one on each side. What causes this? You might be going, wow, this is really weird. Why does this occur? Why aren't these things connecting? What's going on there? Is there something pushing against them, not letting them stick together? No, actually it's due to our Hox genes. Or homeobox genes is what we call them, and they're important for the kind of separation, orientation, and location of different structures, such as our thoracic vertebrae should be above our lumbar vertebrae, and our cervical vertebrae should be above our thoracic vertebrae. They do all the orientation and location, what should be found sort of where, and our paramesonephric ducts should be found close midline together. When they're not and they're separated, Hox genes have failed to bring those two together, and so that's due to Hox genes. Unicornate, this means we only have one half of the uterus there. Obviously, that means one of the ducts, one paramesonephric duct failed to develop. If we have bicornate uterus, then this is just incomplete division. So failure of fusion means that we have two uterus, which is diphallus, and if we just have like two lobes of this, where we have this kind of septum, that's just incomplete of that paramesonephric duct. Okay. Always remember that for females, I know this is kind of a side junction to urinary tract, but females have a shorter urethra, so they're more prone to urinary tract infections. All right. Well, thanks for watching. Don't forget to subscribe. Check out our Facebook page if you're looking for those PowerPoints. And I hope this has helped you for pelvis and perineum and the development of your of the reproductive tract. Check out our other videos. Coming on abdomen is next.